Welcome to a presentation on the biological amendments to Martian Regolith to support successful plant growth. You can't grow plants on Mars. Nutrient deficient, rocky and compact. There's low light and it's really cold. But what if we could amend what's already there just enough to convince a few key crops that they can make it? My name is Angela Ponsano and I'm a sophomore at South Seattle College. I and my research partner, Leah Valentine, a junior at the University of Washington, have been exploring this question for the past year. This research addresses some of the select impediments to establishing renewable food systems on Mars. We're focusing on amending the physical and chemical growth impediments and anticipate that this early work will support research taking into account severe cold and perchlorate levels. Survival on Mars necessitates a self-sustainable system capable of processing and renewing minimal resources available. Martian substrate is dry and compact. These physical qualities would deter plant establishment and impede ongoing growth. Any amendments transported from Earth should be low mass and capable of working with the pre-existing substrate. For this research, we've been working with the simulant Martian Regolith MMS2. It's created by the Martian Garden and is sourced from the same iron-rich Mojave Desert basalt deposit used by NASA to test their equipment's exposure to the unique composition of Martian ground cover. This substrate is designed to be over 90% similar in chemical and textural composition to Martian soil. In this presentation, and the one following, we will demonstrate how plant life can establish in this regolith and growth can be enhanced by the development of the rhizospheres using mycorrhizal fungi and microbes. Last year, our foundational research for this project looked at different ratios of earth compost to Mars regolith, as well as a variety of different plants. As one would anticipate, the more regolith in the blend, the weaker the plant, as we saw plant growth tended to decrease in experimental pots with higher concentrations of MMS2. My research partner and I wanted to see how mycorrhizal fungi and microbes could improve a plant's ability to thrive in a blend with 70% Mars regolith. The microbes we sourced are from Biodyne Inc., an Indiana-based company that generously donated their product to our research. Details on this substance and the impact of microbes will be covered in depth in my colleague's presentation coming up next. The mycorrhizal fungi we worked with is wild root organic concentrate. This product was selected for its blend of both ectomycorrhizal fungi and endomycorrhizal fungi, as well as its low mass due to being a lightweight powder. The mycorrhizal fungi are the fascial network of our planet's vegetation. These organisms strengthen plant roots and help anchor plants into the soil. Their own slender roots are finer than that of a plant, which enables them to grow into the plant root and deliver nutrients more effectively than the plant root alone. It was my hope that while the ectomycorrhizae would physically strengthen the plant's root and allow it to further branch into the regolith, the endomycorrhizae would facilitate increased nutrient uptake. We worked with rosemary due to its ability to network with the mycorrhizal fungi and to withstand both drought and cold climates. Nursery pots were filled with a 70-30 Mars to compost blend and were treated with an NPK fish fertilizer and vermiculite, and this composition was used as our Mars control. Pots were treated with Biodyne, mycorrhizal fungi, and a combination of these amendments respectively. Seeds were planted directly into the substrate, and water was provided frequently enough to keep the soil damp. All pots were confined to a lidded container and treated with 12 hours per day of LED grow light strips with a spectral wavelength of about 45% that of the Earth daylight. As I'm sure you can all relate, we encountered a number of challenges in the fall quarter due to the pandemic. We did our best to monitor and maintain our research subjects, but few survived. My earth control didn't even sprout. However, letting the experiment run wild for a few months allowed for some rather significant results to develop. Of the 30 seeds I planted, only 4 survived: 1 Mars control, 1 Biodyne, and 2 grown in the mycorrhizal fungi. This distinct root growth indicates a successful fungal network that appears to have supported more robust plant growth. These plants appear to thrive, and in distinct ways. The Mars control grew a taproot and a delicate array of branch roots, and a strong pliable stem. The plant grown with Biodyne microbes had almost no taproot, possibly due to its having been grown from a cutting rather than seed, and yet grew long roots and a straight, stiff stem. One of the plants grown in the mycorrhizal fungi, however, was clearly the most successful. The taproot was longer and thicker than the control, the branch roots were longer and thicker, and it developed into a massive network that filled the pot. The plant's height was taller and the stems supple and strong. The leaves were thick and dark green. All of these plants showed sign of woody secondary growth, suggesting that they were well established. The results of these survivors informed our subsequent research. I wanted to recreate these results and address some of the things that may have interfered with the low survivability rate. The fact that the Earth control hadn't grown at all when one Mars control seemed to establish successfully suggested that our low growth rate may not have been due entirely to the harsh regolith. My research partner and I set up matching experiments, arranged to test the impact of microbes and mycorrhizal fungi on plant growth in Mars regolith. Leah worked with experiment A while I attended experiment B. Due to the limitations of maintaining this research at home, our subject number was limited, so having matching experiments expanded our data set. The reset allowed us to address low growth rate. Rosemary is notoriously difficult to grow from seed, so in order to confirm seed viability, we pre-sprouted over 100 seeds in water or iodine-treated water. Once root and cotyledons were confirmed, seeds were transferred into assigned nursery pots. As cotyledons were already present above ground level, the growth rates were tracked immediately. 57 days into the project, the impact of the mycorrhizal fungi on regolith density became apparent. Here we can see a comparison of pots with and without the mycorrhizal fungi. Please note the pockets of space in the plants on the left where the mycorrhizal fungi was not present, compared to the more uniform and fill and minimal pockets of space in the pots on the right where the fungi was added. The packing characteristic of Mars regolith, due to its clay-like consistency, was observed where the surface level had dropped 1 to 2 centimeters over the course of 100 days in non-fungal containers. In containers that were populated with mycorrhizal fungi, the surface level barely receded at all. Pre-sprouting did increase the survivability rate. However, this did not establish success for all the pots. The biodine fungus plants grew in experiment A, but not in experiment B. Mars control plants grew in experiment B, but not in experiment A. And those that did grow did rather well. This suggests that given the appropriate alleles, it is possible for some of the given species to succeed in this environment. All regolith treated with mycorrhizal fungi developed a more loamy consistency regardless of plant success. When considering the survivability, we noted that 70% of all the seeds treated with biodine maintained growth throughout the experiment. Comparably, 67 of the Earth control seeds made it. Overall, when treated with mycorrhizal fungi, more plants tended to survive than those that were grown in just the Mars control. Looking at the final growth recordings of stem height and root length, plants treated with mycorrhizal fungi achieved root length nearly as long as that of the Earth control. When comparing the fungus-treated rosemary with the Earth control, we get a rather distinct sense of the impact the mycorrhizal fungi can have in a plant, even in the harsh environment of a Martian substrate. We've seen in a previous iteration of this work that this treatment will enhance the overall growth in a plant, and in this early stage in the plant's growth, we can see clear evidence of a living fungal network establishing a healthy rhizosphere. In our leaf count comparison, we see typically higher leaf count with amendments, most consistently when mycorrhizal fungi are included. One of the main takeaways of this research is that 3 separate life forms were established to survive in MS2. Mycorrhizal fungi and microbes are essential components to establishing a renewable food source, and each are clearly capable of living in the regolith. This sets an exciting stage for research in the future as we explore how to work with perchlorate levels and the low temperatures found on Mars. The impact of both microbes and mycorrhizal fungi on a plant's ability to thrive in a Mars regolith simulant inspires ideas to take this research further. Increasing the test subjects will provide a wider scope of how these additives influence plant growth in MS2. Biodine Inc. also creates a nutrient pack designed to jumpstart their microbes, which we anticipate may facilitate rhizosphere development more effectively than a standard liquid fertilizer. And pH adjustments with nitric acid and hydrochloric acid have shown promising results, and we would like to see the impact of incorporating these as additional additives. Thank you for considering growing plants on Mars.